record to a cloud. So we're going now, we're recording. These Zoom meetings, these live lectures as I call them, are not supposed to replace you reading the textbook. All right, I have to give that disclaimer almost every day because I know students, they want to do whatever is easy to get the easy grade. These are like almost private tutoring sessions for you to supplement the textbook. It's my job to teach you, to make things easier for you to understand and answer questions and all that bit, but you have to put in your amount of devotion, persistence, and hard work as well. And a lot of that is going to revolve around you just frankly reading the material. I can make it more understandable. I can explain things. I can simplify, but in the next 60 minutes, I can't possibly go through all of the sections of every word in the textbook. So that's always my disclaimer, is that these meetings don't necessarily replace you actually reading the material. All right, so we ended with 4 organic compounds. I think still chapter 2 of the textbook. The 4 organic compounds in the human body are carbohydrates, lipids, proteins, and nucleic acids. We've done carbs and lipids. So now I'm going to go on to proteins, and I'm going to call up a whiteboard so that I can do some drawing for you. So we are on number 3, proteins. The 3rd organic compound in the human body. These are larger. Proteins are typically large molecules. They vary from quite small to huge, but in general, they're larger than most other organic molecules. The building blocks of proteins, I do want you to know this is important. The base subunits or building blocks of proteins are amino acids. This is Bio 1 review stuff. You should have had this in Bio 1. Amino acids are just molecules. There's 20 types of amino acids in the human body. You don't have to memorize all 20 like you may have had to do in Bio 1. These things can act as either a base or an acid, which we went over last time. And they are linked by peptide bonds. So now I'm going to go over the structural levels of proteins. So I will expect you to know this. That's supposed to be an R. Let me fix that. So the levels of structure of proteins, it just goes primary, secondary, tertiary, and quaternary. That just means first level, second level, third level, and fourth level, those fancy words. So in order to abbreviate that, make things go a little bit faster, a 1 with a degree next to it just means the primary level of structure. So the primary level is basically the amino acid chain sequence. So I'm going to write AA for amino acid So whatever the sequence is on the chain of an amino acid, a polypeptide chain, that is the first level of, or primary level of, protein structure. What that means basically is if I have a chain of amino acids, and just for discussion's sake, I'm going to make them different colors, and they're linked together like this. I'm drawing the primary level of a protein structure. And what it means is it's the specific sequence of this chain, black, red, yellow, green, blue. What specific type of amino acid comes next? If I change that arrangement, I've created a different sequence. And so that protein would have a different sequence if the colors were in different order, for example. So primary level just refers to the sequence of the amino acid chain. Secondary level is if we take that primary level chain and we do something physical with it, and we create either an alpha helix, which is a coiled chain. So if I take that chain that I just made in primary level and I twist it like almost like a spring, like this, I coil it, that would be a form, it's actually the most common form of secondary level. There's another form of secondary level called a beta pleated sheet. Don't worry, we're not going to talk about proteins that long. The beta pleated sheet is just a different arrangement, and it's if I take the primary level chain again, instead of coiling it in a helix, I just fold it like this. Take that chain that we created in primary level and fold it. So rather than a coil, it's folding. And that would be the basic two choices of a secondary level of protein structure. Just going to call up a new board and go to tertiary structure. So that is just a 3 with a degree mark for short. So third level structure or tertiary structure would be if we take the secondary alpha helix or beta pleated sheet and we twist it and fold it back on itself. And so if I take an alpha helix, there's a secondary structure. Now if I fold it back on itself like this and make a globular structure, just make a glob and hold it together, I'll use a different color, hold it together with covalent bonds, I've made a big globular structure. That's tertiary structure. So for tertiary structure, I'm going to make a globule or globular structure. It folds back on itself and held together. It's supposed to be an H. It's hard to write on this glass surface. It's held together with covalent bonds. Now we have a tertiary level structure protein, and it's a globule. Finally, quaternary structure or fourth level of protein structure. Not all proteins exhibit all four structures, by the way. But some proteins, all we do is we take tertiary structures, several tertiary structures, and stick those together to make a giant protein. So you can get the picture. If I take a tertiary globule and then I take another tertiary globule and I stick those together, sometimes we take another one, stick it together. Now we've created a really super large protein molecule, this whole thing that has quaternary structure. Okay, that's it on proteins. Except I do want to say, sorry, one more thing about enzymes. These are proteins. All enzymes are proteins. And just to remind you what an enzyme is, all of this should be reviewed, by the way, from Bio 1. Enzymes are proteins that catalyze reactions, metabolic reactions. So chemical reactions occur in our cells in our body constantly, our entire lives, constantly. But most of those reactions are very slow proceeding. And without enzymes, those reactions would never occur. They would never complete themselves. So enzymes are critical to the functioning of the human body because they speed up reactions, they catalyze reactions. And they are not used up in the reaction. So they can be used over and over again. Typically enzymes, you don't have to memorize this part of it. Typically enzymes are very small compared to the substrate. So I'm just drawing an enzyme compared to the substrate that they act on, which is usually a large structure that has a binding site specific for that enzyme. So the red would be an enzyme, the blue would be a substrate. When the enzyme binds to its substrate at the binding site, it's called an enzyme substrate complex. Almost always what happens is then that substrate changes shape. When the binding occurs, the substrate does something to change shape, perhaps like this, and then the enzyme lets go, and it can do that multiple. Here's our enzyme again. It can find another substrate and catalyze it to change shape and do something. Now I'm going to go on to our fourth and final organic compound, and those are nucleic acids. So we've done carbohydrates, lipids, proteins, and now fourth and finally nucleic acids. These are the largest organic molecules in the human body of all four. And there are two subtypes. There's two types. You've heard them, you know what they are. The two types are DNA and RNA. So these are the two basic types of nucleic acids. We could do a whole course on DNA and RNA, as you probably could guess. We're just gonna talk about some basic structures of these. So I'm gonna flip to another whiteboard page and just layer on top of this one. If you need me to go back, I'm happy to do it. The building blocks or base units of nucleic acids are called nucleotides. Since I've been using this term building block, so the building blocks of nucleic acids are just called nucleotides. And they're rather large in themselves. From bio 1, if you remember, they're adenine, thymine, guanine, and cytosine for DNA. And for RNA, they're the same, except we substitute uracil it has a different nucleotide. I'm not going to go into just remember that DNA is the double-stranded helical structure. So if we take a single chain that looks like this is a chain of nucleotides that I'm drawing to the left. If I hook two single chain nucleic acid chains together, we form a DNA molecule. It's a double helical form. So I take this structure and I twist it. It ends up looking like this. So it's like taking a step ladder and just turning it, twisting it. That's what DNA looks like. And these bonds, by the way, that between here and here are hydrogen bonds. So they're fairly weak. So this is what DNA looks like. It's a double-stranded helical structure. RNA, just to remind you, is a single strand. RNA just looks like this, half of that ladder. It's smaller molecule physically. All right, so I believe after I just mentioned ATP, which is actually a form of a nucleotide, I want to say a few things about ATP, then we're done with chapter 2. ATP is a molecule provides energy in the human body. This should be somewhat familiar to you. It stands for adenosine triphosphate. The ATP is just an acronym for adenosine triphosphate. If you have a question, just fire away. You can either unmute yourself or you can type me a question anytime during this and I'll answer, okay?
I have a question.
Sure.
In terms of DNA and RNA, do you want us to know, I know in the book it has like the functions of it. Do you just want us to know that as well? I know you.
That's a good question. So for this first, I get it, that there's so much information in these first 3 or 4, especially these first 3 chapters. You're probably, as a student, all of you are probably wondering, gosh, what's he going to expect us to know for this? It gets really detailed. For this first quiz, I'm going to primarily focus my quiz questions on what I'm, first of all, the sections that I've written in the outline specifically, and what I'm mentioning, talking about in these lectures. So that's a long answer to your question. I'm going to get to the main function of what I want you to know about DNA and RNA before we leave today, okay? It's going to be applied, the function of DNA and RNA is going to be applied directly to protein synthesis. That's what I'm going to spend 10 minutes going over before we leave today. So back to the ATP molecule, it stands for adenosine triphosphate. It's the primary molecule in the human body that provides energy. Okay, so I'm going to quickly draw an ATP molecule. It has 3 phosphates attached to a ribose sugar, which is a 5-carbon ring molecule with a little oxygen right here. I'll label this. This is actually the ribose. That's a sugar. These are 3 phosphate molecules linked together. That's where it gets its name, triphosphate. And finally, there's an adenine group attached to this sugar. And that's a 5-sided carbon. You won't have to draw this on the quiz, don't worry. I just want you to recognize there's the adenine group. It's a 6-sided carbon attached to a 5, which is then the ribose sugars in the middle, and then 3 phosphates out here on the end. Here's how ATP basically works. Whenever we cleave, whenever the human body chops off one of these phosphates, that phosphate molecule gets transferred to something else, and in that transfer, energy gets transferred with it. So this whole molecule, ATP, our cells synthesize it. You know that from Bio 1 with cellular respiration and even glycolysis, our cells make ATP. What I want you to remember is every time we cleave or chop off one of these terminal phosphates, energy gets transferred to something else. That's how this molecule harnesses energy. It's like fuel for the body. What we're going to learn later when we study how a muscle contracts is this terminal phosphate from this ATP molecule gets chopped off and gets transferred to something else in the muscle cell, and that gives it the energy to pull or shorten during contraction. So that's really what I wanted you to remember about ATP. It stands for adenosine triphosphate. It has 3 phosphates. When we cleave this last phosphate off, it becomes ADP, adenosine diphosphate, because it only has 2 phosphate molecules left. We can do that again. We can chop off the terminal phosphate from ADP, and it gives energy as well. And what we're left with then is 1 phosphate on this molecule, and we call that AMP, adenosine monophosphate. So it just goes triphosphate because it has 3 at the beginning. If we chop one off to give energy, now it's diphosphate. We chop one off, now it's monophosphate, AMP. All right, I think that concludes chapter 2. Now I'm going to lead through chapter 3, and I'll be very specific about what I want you to know from chapter 3, okay? So I don't have time to, I'm going to get back to, I don't have time to go through and read you chapter 3. What I do have time to do is say focus on these things for studying for your quiz, because all of this should be review for you. Some A and B instructors, they walk into class the first day and they say, I just expect you to know chapters 1, 2, and 3, and they don't go over it at all. I don't think that's really fair to the students. So I will expect you to know sections 3.1. It just talks about the cell theory. Don't get hung up on that. I might only ask one question. Plasma membrane, which is section 3.2 in your book. I'm going to call up another whiteboard because I am going to expect you to know how the plasma membrane is arranged. The plasma membrane is just the cellular, sort of a cellular wall in a eukaryotic cell, if you remember that business. So let me draw, if we have a cell and its plasma membrane is just that barrier around it, if we magnify that, this plasma membrane is called a phospholipid bilayer. The little head on the balloons that I'm drawing is the phosphate, and the tail, the string, is a lipid. And there's actually 2 of these strings attached to each phosphate molecule. And it's called a bilayer, so we make an mirror image of it like this. The phosphate molecules are the round circles, so that's a phosphate. These tails are the lipid. The phosphate is hydrophilic, that means it likes water, but the lipids, you know, are hydrophobic. They repel anything that's water soluble. All right, so that's how that's the construction of this plasma membrane all the way around. Okay, and there are things stuck in this wall, like membrane proteins. Some proteins are transmembrane, they're integral, and they go all the way through this. So yeah, I'm going to draw a magnified here. This would be a transmembrane protein, like this. And I've drawn it right here. There are others that are just peripheral proteins that sit on the edge, like this. These proteins that are part of this, or they're integrated into this plasma membrane, have several functions. They can act as communication ports, they can act as channels, transport mechanisms. I don't have time to write all of their functions. They act as ways for things from the outside to get in and things from the inside to get out of this cell. That's a very complicated topic. These proteins can just, if they're channels, then they sort of look like this, where things can just passively move through them. Or they can be what you and I call pumps, in which they need ATP or energy to drive a pump and grab something on the outside of the cell and pump it through the protein and spit it out on the inside of the cell. So read about the plasma membrane proteins really quickly. The word glycocalyx, don't worry about that so much. That just talks about the glycocalyx just consists of a protein with a sugar on it. Glyco always means sugar. So a glycocalyx would be a protein with carbohydrate stuck to it, like this. Cellular junctions. If you just look at figure 3.5, you don't have to read all of the fine nitty-gritty about cellular junctions. If you just look at figure 3.5, it will tell you the basics of the three types of cellular junctions, which are tight junctions, desmosomes, and gap junctions. If you just read those little descriptions, that will be good enough for cellular junctions. Layer on top of this, the word diffusion. Now I'm in section 3.3 now, which is just talking about transport of molecules from one side of a membrane to another. This word diffusion is a generic word, which just means the movement of molecules from a higher concentration to lower, and it's passive. It requires no energy. So whenever something diffuses in the body, we don't have to expend any energy for that to happen. So it's just when things move down their concentration gradient. If I have particles, let's say in the air or even in a fluid medium, and they're densely packed here, due to random movement of molecules, they will diffuse throughout the room. It's like if you walk into a room and you uncork a bottle of perfume, that perfume, those molecules will eventually permeate throughout the whole room. It takes a while, but they move passively without the need of energy to where they are of lower concentration. That's a definition of diffusion. Simple diffusion is just what we've described. Facilitated diffusion just means it has help of transport across a membrane. This word, just to remind you, osmosis is basically just the diffusion of a liquid, usually water, a water-based liquid in the human body. So osmosis just refers to the passive movement of liquid or water within the human body. What we're working up to, eventually this chapter works up to, is this. The reason we talk about diffusion and osmosis and all these things is because if this is a cell in the human body and this is the external environment of the cell out here, things move through that plasma membrane selectively all the time. That means some things can go through this membrane and some things can't, as well as fluid. So not only solutes can move across this membrane, but solvents can as well. And in the human body, solvents are typically this, just sort of some sort of water-based media. Tonicity, I'm just going through chapter 3 right now. Tonicity refers to concentration of a solute in a solvent. And this should also be somewhat familiar to these words. If it isn't, if we have a membrane, a cellular membrane that's separating one side from the other, let's just say this is the inside of the cell, this is the outside of the cell. And we have fluid, a water-based fluid, on both sides of this membrane. This is how cells usually exist in the body. And we have things dissolved, solutes dissolved in this fluid on this side of the membrane, as well as this side. Tonicity refers to the concentration of solute and solvent on this side compared to this side of this membrane. That's what that word means. And we use words like hypertonic to describe the side of a membrane that has a higher concentration of solute in the solvent. So I've drawn this right side as hypertonic compared to the left side, which would be hypotonic. Hyper means more, hypo means less. So this side, the right side, has more particles dissolved in that fluid than the left side. This word isotonic just means if the concentrations were identical. So isotonic would be if the concentrations of solute in solvent were identical and the net flow of materials would be even. The last thing I'm going to say about tonicity is this. Fluid, water, always moves toward the hypertonic side of a membrane. Fluid always wants to move toward hypertonicity. And if you remember that, you can answer any question about tonicity whatsoever about the movement of fluid. It always moves toward the hypertonic side, or wants to anyway. The next section is in your textbook is just about active membrane transport. That is opposed to simple diffusion or passive transport. So active membrane transport I'll just ask you a couple questions on the quiz from this as well. This requires, first thing, whenever you see this term, active transport, know that that process requires energy in order to proceed. And usually in the form of ATP. ATP gets expended or used during this process of active transport. I also tell students, think of it as an active pump that requires energy. So I'm going to give you a quick example. If we have a protein membrane pump that sits on the cell like this, this active transport pump has the ability to pump certain things out and other things into the cell. When we do the nervous system, which is last in this course, we're going to talk about the sodium-potassium membrane pump, which is the most common membrane pump on our cells in the human body. And what this pump does, it's ATP driven, requires energy, and it keeps pumping sodium ions outside of the cell and potassium ions in. And it pumps them against their electrochemical gradients. That just means even though all ions want to equalize across this membrane. So sodium wants to constantly move into the cell and potassium out, but this pump works against that desire for this to equalize, and it keeps pumping this sodium ions out and potassium ions in, and in the process requires ATP or energy to do so. Ultimately, what's going to happen is because we increase the concentration of sodium out here, potassium in here, what we're creating is a polar environment where we have positive out, negative in. That's going to give this cell the ability to do energy. I'll explain all that when we get there. In the nervous system, mostly. Vesicular transport is not that big of a deal. I'm in section, I think 3.4, I believe at the end. After active transport, I know I'm going fast through this. It's because it should be review from Bio 1. I'm just going to mention these things in A&P during a process of like phagocytosis or exocytosis, endocytosis. Basically, vesicular transport just means the movement of something inside of a vesicle. And a vesicle is just a bubble-like structure. So I'm going to give you some examples. Here's a cell plasma membrane. If this cell wants to engulf something inside of it that's out here, outside of the cell, it can do so by engulfing these particles into and creating a vesicle. Phagocytic cells do this all the time. Well, those are immune cells, really. So let's just say this is bacteria. These things are bacteria or virus that's gotten inside of our body. We have specialized cells in our body that can engulf this foreign microorganism, internalize it, and then once it's internalized, break it down or destroy it. That's an example of vesicular transport. Likewise, it can take the debris of the broken down microorganism, package it in another vesicle. Let's make it a different color now. So this is the debris of the broken down microorganism. Now this cell sometimes has the ability to get rid of that debris also through a process of exocytosis. So it spits it out of the cell. These are just examples of vesicular transport. Engulfing here, transporting in a vesicle, breaking down, and then exocytosis of the debris. I believe on my outline, I skipped section 3.5, selective diffusion, roles of CAMs or cell adhesion molecule. Part 2 in chapter 3, which is starting with section 3.7. I'm going to be very specific. I just want you to go through the organelles and know their basic functions. It's not going to be a major part of the quiz. By the way, while I'm thinking about it, these quizzes are usually anywhere from 35 to 50 questions, depending on how much material we have covered in that time. They're all true, false, multiple choice, if you didn't hear that yesterday. And what I'm going to do in this section of the quiz, when I'm just going to ask you simple questions about organelles and their functions to see who revisited this in the textbook. So quickly, I'm not going to write all this stuff out. I don't have time because I want to get to the question about DNA and RNA. So quickly go through your book and just write down each organelle. I would make an outline. Mitochondria. Number 1, that's the site of cellular respiration. That's where our cells combine an energy source like glucose and oxygen to make or create, synthesize ATP. So it's a place of oxidative phosphorylation. So it's a place where we use oxygen and an energy source to create ATP. That's why sometimes it's called the powerhouse of the cell. Ribosomes are these little tiny organelles in the cytoplasm, and they are the site of protein assembly, where proteins are actually put together or synthesized. Endoplasmic reticulum is a membranous structure, and it's encompassed in these things called cisterns, coiling all around. It's usually associated with the nucleus, and there's 2 types of endoplasmic reticulum, rough ER and smooth ER. That just refers to rough is, does it have ribosomes on it? Know that if it has ribosomes on it, if it's rough endoplasmic reticulum, it's associated with protein synthesis because ribosomes are where proteins are synthesized. So rough endoplasmic reticulum then is typically responsible for packaging the proteins that are made on those ribosomes and packaging them either for export through the Golgi apparatus or to stay within the cell. Smooth ER, which does not have ribosomes on it, has all sorts of functions. It's usually associated with some sort of lipid manufacturing. It metabolizes lipids, it makes cholesterol and phospholipids for the membrane, synthesizes steroid-based hormones from cholesterol. It has something that smooth ER almost always has something to do with cholesterol, lipid, fat, those types of things. The Golgi apparatus is a packaging center usually to export proteins outside of the cell. Peroxisomes are just sacs that detoxify the cell. So inside of a cell, for example, if I have this little vesicle, a peroxisome, this is a site, a center to detoxify toxic chemicals, things like poisons that get into our body, alcohol that you drink, that actually is poisonous to your cells. So that gets broken down in peroxisomes as well, things like hydrogen peroxide. A lysosome is like a peroxisome, but it acts more like a recycle center. So think of peroxy as a detox center and a lysosome as a recycle center. It breaks down not just foreign material, but worn out particles within the cell. It's like a trash compactor. It breaks them down and then recycles some of those pieces. Okay, now last but not least in chapter 3, I'm going to skip to this is what I want you to know about DNA and RNA. I'm going to clear up my board here so that I can add something on. I'll write this. Section 3.11, RNA, DNA in the context of protein synthesis. I will ask you a few questions about this process, okay? And before we have the quiz, probably tonight or tomorrow, I will try to post some sort of another outline for you to help you study for the quiz on our Canvas web page. It'll just be basically a review of the outline that I've already posted, but I'll try to highlight more specific things like this. So what we're going to do is we're going to make a protein. I'm going to explain to you in the last 10 minutes of this lecture how a protein is manufactured in a cell. First thing I'm going to do is draw a really big nucleus within the context of a cell. Most of you know this from Bio 1, at least you should. What's contained in a nucleus is our genetic information, DNA. So I'm going to draw a DNA molecule in here. There it is. And it's held together by these little hydrogen bonds. It's coiled, double-stranded, and then twisted, coiled DNA. The first thing that happens during protein synthesis in a cell, and the reason I'm making a big deal about this is because that's usually most of what our cells do all day long, is manufacture proteins in some form or another. This DNA, this molecule, this genetic information, it's the software. It holds the code for all the proteins that need to be made in our bodies. So this is the software or code, and it's in sections called genes. So a gene is just a section of DNA that holds the code for specific protein. Now, the first step that happens when a protein is synthesized or need to be made is this DNA molecule gets signaled to uncoil. So step 1, I'm going to draw this. This DNA molecule, it uncoils. And these little protein structures called histones that hold this molecule together, I'll draw those in red. What helps hold this DNA together in this double helix structure are these little red proteins called histones. Those histones from this signal the signal that came from outside of the nucleus, the histones are temporarily removed in this part, and the DNA uncoils. Now, The next step is a single-stranded, let's just say, here's the gene, the software of the protein that we're going to make. There's the code, the software. We opened up the DNA molecule so that gene could be accessible. It's not tightly coiled anymore. It's physically accessible so that we can make a copy of that gene. To cut to the chase, I'm going to draw another picture in a minute. The ribosome, where the protein gets put together, is out here in the cytoplasm. That's where we assemble a protein. This thing in here has the code for the protein that needs to be made. But DNA is too big to get out of the nucleus. It can't do that. So what we're going to do is we're going to make a copy of this gene, a copy of this code that's in yellow, so that that copy can get outside of the nucleus. And we do that in the form of an RNA molecule. So an RNA molecule is single-stranded, and it's made from this DNA template. So what we do is we copy, just like a photocopy, of this yellow circled code. So the DNA, just to blow it up for you, the DNA looks like this. It has a code to make the protein on it, but it can't leave the nucleus. So what we're doing is we're making a copy of that code in an RNA form. RNA is single-stranded, and RNA can leave the nucleus. Now here's the catch about this RNA molecule. This code from the DNA has a lot of gibberish on it, a lot of nonsense that doesn't make sense. So the RNA first copies everything on the DNA. So everything in green is a copy, but there's a lot of gibberish or nonsense on this copy from the DNA code. We need to get rid of the nonsense and keep the stuff that makes sense. So we process this RNA molecule. That word, processing of RNA, just means getting rid of the nonsense. Okay, so we're going to process the RNA, chop out all the pieces that don't make sense. Like, let's say that piece doesn't make sense, that piece doesn't make sense. We're going to get rid of those and then sew it back together. Okay, so we process the RNA and it becomes now messenger RNA. So I'm going to put a little M, RNA. So we've processed our original RNA, gotten rid of the nonsense. Now we have mRNA, and this leaves the nucleus because it's small. It has the code for the protein on it. We're just transferring the code that was originally found on this DNA gene. So we've made a copy of it, we've gotten rid of the nonsense, it leaves the nucleus. Now this mRNA has the code to make a protein. That has to happen out here at the ribosome. The ribosome is the assembly plant for a protein. Now I'm going to go to another, I'm going to layer over this, clean up my drawing a little bit. I'm going to shrink my nucleus because we're not going to talk about that anymore. Now we've got our messenger RNA, and it literally, like ticker tape, goes through the subunits, the ribosomal subunits like this. And the code on this messenger RNA is read. The ribosome reads the code in triplet base pair nucleotides. So every 3 nucleotides is a word on this messenger RNA, and the ribosome reads these triplet base pair words. It would just be like if the English language contained all 3-letter words. Obviously that sounds ridiculous, but that's how the language of RNA of genetic information is read in triplet base pair codes. So they're just 3-letter words. And as they go through the ribosome, they are translated by the ribosome. So the process of translation occurs out here at the ribosome, and that's just the reading of this message. What we did up here by making an RNA copy is called transcription. So transcription is the making of a single-stranded RNA from the DNA template, the master code. Translation is the deciphering of that code on the messenger RNA at the ribosome. When that coded message is translated or read, it signals a specific amino acid out here in the cytoplasm. I'm making them different colors. So these are all amino acids out here in the cytoplasm of the cell. As this message gets read here, it signals a transfer RNA molecule, which is just a different RNA molecule. I'm going to make it look like a T. This message right here says, bring this. It signals the transfer RNA molecule to bring its specific amino acid and deliver it to the ribosome. So the first 3 letters in this coded message say, bring the blue amino acid. I'll label this transfer RNA. And each of these amino acids has a transfer RNA. They're just shuttle buses, transfer RNA molecules that bring amino acids to the ribosomes. Then let's say the second 3 letters in this coded message says bring the red amino acid. And so the transfer RNA molecule, the shuttle bus, brings it into the ribosome and it hooks up the red amino acid to the blue one. And then let's say the third, I should make that a different color. The third letter that is read on this long chain, let's just say it bring the black one in. Transfer RNA molecule shuttle buses that black amino acid and hooks it up to the chain. And finally, the fourth coded message, triplet base pair code, says bring the green one. Now we've created a chain of amino acids. Guess what that is? That's the primary structure. I won't use green because then it might just confuse you. Remember, the primary structure of a protein is the sequence of amino acids that got made. And that's this particular protein primary structure is blue, red, black, green. That's a sequence. That's the primary structure. That then dictates secondary structure, tertiary structure that we just learned. So really quickly in the last 2 minutes of this lecture, what I'd like you to know about protein synthesis is that transcription is the making of a single-stranded RNA molecule from the DNA template in the nucleus. Then that RNA molecule gets processed in the nucleus. And the processed RNA molecule is called messenger RNA. That's little m RNA. It leaves. The messenger RNA travels to the ribosome out here in the cytoplasm, where that messenger RNA is read or translated in triplet base pair coded message. It's the triplet base pair code that dictates which amino acid is shuttled to the ribosome based on the code by a transfer RNA molecule. And these transfer RNA molecules, they just bring an amino acid in, hook it up to the chain, and then they go fetch another one, should they be signaled to do so. So that's what I want you to know about DNA and RNA for this first section, because this will come up later. When we get to the end of this course and we learn about steroid hormones, what hormones do, that's going to be, a steroid hormone is actually going to be the chemical signal to start this whole process. All right, I'm going to be done for the day, so let me shut off my recording real quick, and then you can ask me any questions you like.